Hi, my name is Dina Newman, and I am a faculty member here at Rochester Institute of Technology in the School of Life Sciences. I'm part of the Biology Education Research Group, and I'm excited about the opportunity to mentor some new students as part of our DBER REU this summer. My main interests are in the area of how students think about cellular and molecular biology and how representations of molecules and processes affect their thinking. I work very closely with Dr. Kate Wright, and we've had a team of undergraduates working on several projects, including student conceptions of meiosis for the past few years. Meiosis is notorious among faculty as a topic that students have difficulty with, and if you're not remembering what meiosis is, here's a sort of typical representation. It's cell division that results in gametes, starting with a diploid cell, separating the homologous chromosomes, then separating the sister chromatids, and ending up with 4 gametes. This is a very important topic because it explains how information is passed down from one generation to the next. But we have found that students typically do not even think about information when they're thinking about meiosis. They find the process very confusing, and we categorize their confusion into 4 main categories: chromosomal structure, homology, ploidy, and molecular mechanism. To make matters worse, we find that the concepts that we consider foundational to these 4 areas are often not even mentioned in introductory level textbooks. So we hypothesize that active learning exercises focusing on these 4 areas would help alleviate the problems that students experience in learning about meiosis. I would love to have an REU student continue working on how to solve this problem. Another branch of my research started with student interpretations of central dogma, which is often depicted like this: DNA, arrow, RNA, arrow, protein. It's another essential topic that falls under the heading of information flow, but which students do not think has anything to do with information. One of the confusing things about this representation is it kind of looks like a chemical reaction. It looks like DNA is being converted into RNA, RNA is being converted into protein, which of course is not at all what this is about. So that got us thinking. Chemistry uses a lot of very specific kinds of arrows for very specific reasons. But what about biology? So work we've been doing shows that there are dozens of different arrows that get used in biology for dozens of different reasons. The same arrows are being used for different purposes and vice versa, and very often even inside the same figure. So we think this must be very confusing for someone trying to learn biology. Again, I would love to have an REU student continue that project. I'm also working with Dr. Wright on 3D physical models of molecular concepts, and with Dr. Baptiste on developing modules for interactive learning of biology concepts, which includes making interactive video vignettes. You can watch Dr. Wright's video to learn more about those projects. Thank you for your interest.